Hi everyone, I'm Ramkumar Mathur. I'm Department of Geriatrics. I'm assistant professor over there. So thank you so much for TRANSCEND team to giving me this opportunity. So I work on basically on immune aging and mostly on basic research, mouse work. And so this project is in collaboration with Dr. Stika Sanford Clinic. So he provide like sample for this. So mostly we are looking like CD8 T cell, how they are the transcriptional profile in the tumor microenvironment, how the tumor microenvironment actually affect the CD8 T cells transcription. So let's move on. So CD8 T cells are very critical to the tumor immunology, and as you can see, like here CD8 cytotoxic CD8 T cells are interact with the tumor cells, and they secrete to granzyme and some lytic enzyme, and which kill the tumor cells. And in the so tumor actually make some evasion mechanism, and they express PD-L1. It's called like program death ligand, and this bind with the program death receptor is called a PD-1, which is expressed in the CD8 T cells. And that's how this interaction, the PD-L1 and PD-1, which is like pretty established in the tumor immunology field, and this interaction leads like CD8 T cell exhaustion, and the tumor cell are evade from that immune check from the CD8 T cell. So CD8 T cells are really critical for the tumor immunology. And as you can see over here, as we age, like an aging immune system actually decline, and they have more intent to tend to like evade the tumor cells, evade like from the CD8 T cells killing. And so the question is like how this CD8 T cell epithelial immune interaction shape the tumor microenvironment, and like that leads to metastasis and tumor resistant in aging. So what are the underlying mechanism for that? So we found like a pretty like interesting mechanism, like how this epithelial cells, like this is like a fucosylation, which is a post-translation modification, and it's kind of like fucose adding to the glycoprotein and oligosaccharides. So these are different kind of type of fucosylation is happened by with some fucosal transferase. You can see like multiple type of the enzymes over there. So in steady state, the fucosylation is really required, which actually like help for the to maintain gut homeostasis and protect from the pathogenic infection. Although what we found that in the aging condition, so we compare the young and old, and on the basal level, the fucosylation is higher in the epithelial cells of the old people. And what we found, like interestingly, like you can see, like if epithelial fucosylation is higher in the tumor condition when you compare with the young versus old tumor, and they are pretty significantly higher. And as you can see with stage also, the older people have stage compared to young, they have a higher fucosylation. So saying that this fucosylation is related with chronic fucosylation is related with colon cancer risk in the older adults. Then we look at like a more deep inside how this fucosylation, what is the molecular mechanism, and they look at like single cell RNA-seq, and so we took like a young and old. So this is like a study, we analyzed the single cell RNA-seq, and we found like this fucosylation is pretty like predominantly express more epithelial cells. This is the colon, this single cell RNA-seq. And in particularly you can see like a futate, or like they are like differentially expressed in the young versus old like tumor, and this futate is like mostly like the predominantly in the old tumors. So and apart from this, we look at like this in single cell. So we look at the epithelial cells and T cells, and found this epithelial cell has like more PDL expression in the in the old tumor condition. So and also like the, I'm not showing here, like the PD-1 also expressed more, and like in the CD8 T cells. So we further validate the finding in the mouse. So I use this pretty established model like AOM injection and multiple round of DSS, dextrose sodium sulfate, to the mice, and the mouse developed the colon cancer. So what we found that compared to young, the old epithelial cell has a higher epithelial fucosylation as we found in the human condition. Interestingly, like what we found, like when we take the CD8 T cells from this tumor, and then we found like PD8 expression is higher as compared to like tumor in the young. And also like the very interesting finding we found, like this also this cell has the more P16, which is like pretty like a obvious, like because you can say like old has the more like a P16, but like what interestingly I found, like in the young, like in the tumor condition, they have more P16 marker compared to the uncontrolled. So which is telling like a so there are like certain report like the PD1 therapy is actually like the getting like a failure. So maybe like this like the CD8 T cell also like get like the senescence as well. So we hypothesize whether a dysregulated epithelial fucosylation reprogram the tumor microenvironment and this stabilizing the PDL expression, and which enforcing CD8 T cell exhaustion and senescence, and maybe this is driving the metastasis. So we test our hypothesis in the two aims. So first aim, we look at the transcription for profiling of CD8 T cells in the tumor region. So we hypothesize that look at the T cell, T cells from the fucosylated region and non-fucosylated region. So our hypothesis is the fucosylated region are having more PDL1 expression, which interact like CD8. So that will lead like a more immune evasion versus non-fucosylated. So there are very nice technique with the collaboration with the genomic core, Damien. I discussed with them, like so we have in-house this in the UND like this nanostring this technique. So what we can do, like we will like stain epithelial cell, CD8 T cell, and like also like fucosylated, and we can make this ROI and then capture this region and like look at like transcription profile epithelial cells and CD8 T cells, and then we can make like nice correlation with that. So I will like do so. I will recruit. We will recruit the 40 patient, 20 young and 20 old with the collaboration with the Dr. Stika in Stanford, and then we like the tumor cells and then the PBMC and make like first like we do the nanostring and look at the nice interaction, epithelial and CD8 interaction and transcription profiling, and look the functional assay with ISC validation with ISC and some functional assay. So this will tell us like epithelial CD8 interaction and distinct tumor gene signature in tumor microenvironment. And in aim2 is like more validation, and then here is what we do, like we will look at like more distinct biomarkers. So we will integrate clinical data from those 20 patients and also like the nanostring and transcriptomic and ISC like this, and use some machine learning approach. And what we will do, like we will find like network analysis and the special biomarker. So we understand like these are the less for making this training model, and we have less sample, but so we will validate further in the available data set in the TCG data set, or in-house we have the established mouse model where we can recapitulate that. So we will look at whatever the markers we have, we can test like those to make like better immunotherapy. So this will tell like the outcome is like identify epithelial CD8 T cell cross-talk for immunotherapy and enable what are the early therapy adjustment how we can make it. And also like as I show you, like P16 was like or also increased. So in the combinatorial therapy with the PD1, how we can like make effective this immunotherapy for the older adult. So here's my team, and like Dr. Stika is like a co-PI, and Dr. Jurbich is like he's a geriatrician, he's assist for the patient certification, and my mentor is Dr. Khushro Rizwani from University of South Dakota. And I always acknowledge the like research design team, Mark, Jungkook, and Laura, and so administrative support like Savannah, Miranda, and Casey like for helping with the IRB and UND Genomic Core and Histology. And thank you so much. I will happy to.
All right, questions?
I have one quick one. Sure. You say in aim 2 that you're going to predict therapy response. these are metastatic patients. How are you going to predict therapy response? What is the outcome you're looking for?
So we are looking for example, like 20 patients, we are looking like the whether these are like so these are not like immunotherapy. So we exclude those like those patients, like we are not like looking like some like immunotherapy like one. So here still we get like some like the signature gene, like those are like the CD8 T cells, like how they are like the CD8 epithelial, like those are like skipping those genes, so we look at that.
So those are surrogates for therapy. You're not going to look at survival or anything like that, are you?
No, like I think mainly like the because this is like pilot of 20 patient, like we can look at more the like signature gene, but definitely in AIM2, like I told you like in the publicly data set, like I will like look at like what are my findings. I will like validate with that, like whether this is true, like how the fucosylated region, like like there is like the correlation, and there I will look at in public data set, like how this work with whether like like the signature gene, like for example, like either like the young versus early onset or like late onset, like whether P16, those aging marker, like senescent marker, and are they are like and PD1 are they are really responsible for that. So we can like predict that whether can we target for the, or already like those people are like on immunotherapy, those data set we can look at that, like whether this the immunotherapy is failed because of the PD1 even like therapy is over there, and whether P16 marker or like these CD8 T cells are getting senescent, acquiring the senescent, even the young early onset. So we can definitely like predict those. Yep.
Thank you.
Yep.
Any questions? All right, with that, let's give another round of applause to Dr. Mathur.
Thank you so much.